So, welcome everyone. The webinar is about to begin. I introduce myself. I am Géraldine Ménien. So I work in France on chestnut sector, and today I am delegate to host this webinar. With me, there is Hélène Lopez from Interco, who will help me in the chat, especially. You may have seen in the introduction that all presentation will be in English. So you can activate subtitle in your language by selecting subtitle in the menu bar. And you can also find the procedure in the chat. I know the translation is not perfect, so I hope you will understand my English and the speaker's English. The scientific word will not be well translated, so thanks for your understanding. So, yes, please, everyone, mute your microphones and camera with the exception of the speaker, please. So the chat is open. All your questions can be left in the chat, and then we will cover them at the end of the webinar. So before I go and introduce the program, I would like to give the floor immediately to Bertrand Guérin. Hello, Bertrand. Bertrand is a chestnut producer in the southwestern France, and he is the president of the Eurocastanea Association that organized this webinar. So the objective of the webinar today is to make the point on what we know about chestnut rot. So Bertrand, you wanted to organize this webinar. Can you tell us a few words of introduction, please?
Vous m'entendez?
Yes, we can hear
you.
you.
Okay. Hello, everybody. Just few words to welcome everybody. As Géraldine said, we have a very important new problem since 5 or 6 years. Maybe in France, maybe in 2018, something like that, following the CNIPS, a new fungus appeared. I don't know who gives the lovely name of this fungus. It's Gnomoniopsis. Maybe it's you, José, or I don't know exactly. But now it's causing very, very important damage on the fruits and the global production in Europe. I don't know if it starts in France, but now everywhere in Europe we have this fungus, and we think we need a collective action to fight and to win the battle against this fungus. Maybe like we did with CNIPS, when CNIPS appeared in 2010 in Italy. I think now the problem of CNIPS is okay. We don't have damage now because we have a very important mobilization of everybody, and I think we need to do the same for Gnomoniopsis. Well, it's not. I think today it's a way to have information of everybody, because everybody working on this problem in Europe, in France, in Italy, in Portugal, in Spain, but also in Chile and in other countries in the world. and it's important to talk about this problem and to try to have a planification of our actions in the future to win this battle. Well, Geraldine, maybe you can present the different speakers now and the different items we have during this meeting.
Yes, so you can see the program on your screen. I would like to thank all the speakers to be here with us. For the first part of the conference, we will ask the question, what are we talking about? We will see what is the problem in Europe with José Laranjo, but José have a last minute problem, so he will come back in 15 minutes. So we will see what happened in the other country, and then we will have the presentation of José. So we are fortunate to have with us 3 people from Australia, Chile, and United States. They will tell us what is the situation in their country. First, I will turn it over to Mrs. Celeste Linde. Celeste is from the University of Canberra in Australia. Hello, Celeste.
Hello.
I think, Celeste, we have about 10-hour time difference. So we are in the middle of the night at your place, if I'm not mistaken. So thank you very much for your connection. So Celeste, please, can you tell us what is the situation in Australia? You can share your screen, please. Okay, that's perfect. We can see it.
All good. Can everybody see? Everybody can hear me?
Yes.
Fantastic. I'll start by thanking you all very much for inviting us here all from Australia to join in this webinar. It's a great honor to participate in this. Sorry, let me just get my... So Gnomoniopsis in Australia has a fairly long history. The industry in Australia itself, the chestnut industry, is not very big, but they have been looking into the nut rot problem for quite a while. So even in the 1990s, it has been identified as a problem causing rots in nuts. At that stage, it was identified as Phomopsis castanea, not yet Gnomoniopsis. And that was by Washington et al. In 1999 when that was published. Subsequently, that fungus was then described in Australia as Gnomoniopsis smith-ogilvii. Now, I know this name is contentious. In Europe, you use castanea. In Australia, we prefer to use smith-ogilvii. It doesn't really matter what the name is. The important thing is that we try and figure out how we can manage and control this disease. So the Australian chestnut industry have been actually quite active in funding research into this pathogen for a long time. So in the 2010s, they funded a PhD project in the name of Lucas Shuttleworth, and he, of course, did some groundbreaking work initially in figuring out the epidemiology of the fungus. Also confirmed that it's an endophyte and looked at differences in cultivars as well. So currently it is the most important disease in Australia, and it's it's a really threatening disease. And again, I'm very pleased that this big drive is currently going on to understand the disease better. In Australia, the incidence vary from year to year, and that depends mostly on the environmental conditions as far as we can figure out. So some years, the incidence is more than 40%. In other years, such as last year, the incidence was very low, only 0 to 5%. And we think it's because last year was a very cool and wet year compared to other years, which is usually hot and dry. In Australia, all cultivars are affected, but bush, the Bettyzac, is probably the cultivar that shows the most rot, and that's also the cultivar that flowers and fruits first. So what do we do in Australia currently for control? We don't really have a lot of options, but there has been some work done by Deakin University in Australia where they've trialed a few fungicides, first in vitro growth, and then in the orchard as well. And they, from the fungicides that they tested, they suggested that pyraclostrobin and difenoconazole, especially in combination, works best. And so currently in Australia, these are being trialed in grower trials, but we would be looking to expand on these trials, perhaps including things like the phosphonate salts or other options as well. So of course, there's still many questions around nut rot, especially around the predisposing factors to the disease, and also still questions about the epidemiology. And it'd be good if we can nut these out a bit more. The chestnut industry, even though it's relatively small, they do fund some projects through a research and development levy. And hopefully in the end, some of this research could be used in, for instance, a disease prediction model, if possible. And that's it. Thank you.
Okay, thank you very much, Celeste, for this presentation. If you can, I hope you can stay with us for the question session. Sorry.
Absolutely, yes.
Okay, perfect. Thank you. Now I will give the floor to Mrs. Viviana Cisterna from Chile. Viviana worked at the Research Institute of Chile, and she will present the situation in Chile. Viviana, I let you speak. You can share your screen, please. Viviana, your microphone is cut. Viviana, we can't hear you.
Viviana, ¿puedes abrir el tuo micrófono, por favor?
¿Ahora sí?
Yes,
Ahora
we can
sí,
hear you.
ahora
Thank
sí.
you.
Good morning. My name is Viviana Cisterna Oyarce. I am a researcher in the Agricultural Research Institute of Chile, and I will talking about the situation of Gnomoniopsis castanea in our country. To put us in context, Chile is a country where the chestnut tree plantations are abundant. The area of chestnut trees in Chile has increased by 500% compared to 2004, reaching almost 1,500 hectares in 2022. The increase in chestnut true area has translated into the exportations. The chestnut exportation reached 4 million kilograms nut in 2023. As you can see in the graph, the main destination markets were Italy, Portugal, and Spain. However, some Chilean exports have recently experienced problems with microbial infections. The brown chestnut exported to Europe, together with the brown chestnut processed in the lab 30 days before arrival, were already 30% damaged at the time of arrival in Europe. They were infected by different microorganisms. However, 85% of the damage was caused by brown rats. The same chestnut, when processed 90 days after arrival, had up to 70% damaged brown rats. In the case of the navy chestnut, the damage due to Gnomoniopsis castanea was about 19%, and many consignments has to be used for animal consumption. In these pictures, you can see the level of damage to the chestnut after arrival in Europe. Looking for a solution to the infection, we start our research. We analyzed a total of 76,000 chestnut fruit of 7 varieties for the evaluation of post-harvest disease during the years 2018 and 2023. This research was conducted in the Applied Microbiology Laboratory of the Agricultural Research Institute of Chile. As you can see in the graph, during the 6 study seasons, Marrone di Cuneo was the variety most affected by Gnomoniopsis castanea. To dive deeper into our research, during 2023, we conducted monoconidial cultures of 45 strains from different regions of Chile and different varieties. The cultures are deposited in the bed of microbial genetical resources. Additionally, during the current season, we have been analyzed chestnut tree structures in different phenological states to identify the presence of Gnomoniopsis castanea. Despite it has been detected in different phenological states in other countries, we haven't found it in Chile. This is the situation in Chile. Thank you very much for your attention, and don't hesitate to contact me if you have any questions.
Yes, thank you, Viviana, for this presentation of the situation in Chile. I hope you can stay with us. Now, I know that José is back, but I propose we conclude this overview of other country with Mrs. Giorgia Bastianelli from Michigan. Giorgia is working at the University of Michigan. Good morning, Giorgia.
Good morning, everyone.
This is the morning in Michigan.
Yeah.
So thanks to be with us. I let you speak about the situation in United States.
Okay. Can you hear me? Can you see my
Yes.
screen?
Yes,
Perfect.
absolutely.
Okay, I'm Giorgia Bastianelli from Michigan State University, from Timothy Miles Lab, the small fruit and hop pathology lab. And today I'll show you some quick updates on chestnut brown rot in Michigan. And after chestnut blight, after chestnut blight decimated the endemic American chestnut tree, Castanea dentata, in Michigan, mixtures of chestnut species have been planted in the current orchard, including some European Japanese hybrids like Colossal and some other Chinese chestnut trees. And Colossal is a very precious cultivar with very large nuts and the greatest yield of any cultivar in Michigan, even if it's not very cold tolerant. And on the other hand, the Chinese chestnut trees are chestnut blight resistant, but don't yield well when compared to Colossal. And in the last years, more or less in the last 20 years, chestnut production in Michigan has had several setbacks, especially because of pests and pathogens, and also pollination and coat issues. And then the IKB, the internal kernel breakdown, which is basically a genetic incompatibility that occurs when Chinese cultivars pollinates the European Japanese cultivar like Colossal. And last but not least, chestnut brown rot has been added to this list recently. So let's have a look on chestnut brown rot history in Michigan orchards. In Europe and Oceania, brown rot symptoms were first detected early in the 2000s, and Gnomoniopsis was identified for the first time in 2012, both in Italy and Australia. And later in the years, Michigan growers didn't find similar symptoms until 2016, even if Gnomoniopsis was isolated in 2012 from chestnut blight cankers. And 1 year later, in 2017, Gnomoniopsis was found in 80% of orchards in Michigan. And then start from 2018, brown rot symptoms affected 6% of nuts at harvest time. And then in 2019, the 80%, and up to 9% in 2020. And for these reasons, MSU pathology lab started investigating about chestnut brown rot. This work was carried out by Ellie Watson, a graduate student at MSU, and she evaluated and compared brown rot symptoms on chestnut from 3 different cultivars in the same orchard, including Colossal, which is, as I mentioned before, the European Japanese hybrid, then Benton Arbor, which is Chinese cultivar, and then Labor Day, a Japanese cultivar. And we can see how in the 3 years investigated, 2019, 20, and 21, Colossal cultivar resulted significantly more susceptible to brown rot, considering both the incidence and the severity of the disease, while the other 2 cultivars didn't differ one from each other. And this is the quality summary of 2022. Carried out by CGI, Chestnut Growers Incorporation in Michigan, in terms of brown rot average in the season. And in 2022, at harvest time, brown rot symptoms were on 6% of chestnut delivered, percentage that increases up to 32% at the floating phase of the treatment chain, and then degrades after packaging to 7%. So brown rot was quite steady between the delivery and packaging. And the last year, in 2023 harvest season, we got similar results about the quality of 63 tons of chestnut surveyed by CGI. And we can see how brown rot wavered between 2 and 9% at harvest time, considering an early or a late delivery in the season. And then again, as happened in 2022, brown rot percentage increased up to 25% in the floater fraction and decreased after packaging to 4%. And in this case, we have a sensitive decrease in brown rot between delivery and packaging. But again, we don't know what happened after long-term storage. So Why should we care about brown rot in Michigan? What does it mean for growers in terms of yield and income? Approximately 12 tons of nuts out of 63 tons delivered in 2023 were discarded before packaging for quality issues. This translated into economical loss means about $100,000 lost before harvest. And moreover, half of the discarded nuts had actually good quality, but were discarded during floating. And about 700 kilograms of nuts from the floated fraction prior to packaging. So what can we do to help growers in Michigan? First of all, it's important to test, and if it's possible, to label new chemicals or biofungicides effective against brown rot in field condition. And then it's also important to test the efficacy of post-harvest treatments to spot critical points in the chain that lead to an excessive discard of healthy nuts and don't prevent brown rot symptoms developing during storage. Pre- and post-harvest strategies are strictly related one to each other. Effective field treatments can reduce brown rot symptoms in field, but if Gnomoniopsis is still present in low percentage in nuts at harvest time, effective post-harvest treatments can inactivate the inoculum and prevent the spread of symptoms during storage. And in the last section of this webinar today, I'll show you what we did to reduce brown rot in Michigan orchard. For now, thanks for the attention.
Yes, thank you, Giorgia. You stay with us for the second part of your presentation about the means of control. That's exact?
Yeah.
Okay, thank you. So, thank you. We understand that the problem of Gnomoniopsis is a global one. I would like now to give the floor to José Laranjo from Portugal. José is a professor at the University of Trás-os-Montes in Portugal. And he will present the situation in Europe. What are we talking about? What is the problem of Gnomoniopsis in Europe? Thank you, José.
Okay.
You can share your screen.
Mm-hmm. Here. Okay. Ready?
Yes.
Okay. My first words are directed to the collaborators from the other countries that helped me to prepare this presentation about, well, an overview about the Europe situation. So, presentation mode. Okay, good. Okay, well, I started this preparation a few weeks ago, sending an email to many colleagues from other countries, asking them about these questions, as you can see. Well, about the Gnomoniopsis, the first symptoms, and as we know, Gnomoniopsis is very well connected with Galloasp. I also asked them about the presence of the Galloasp in their countries. Well, I have answers from Austria, France, Georgia, Greece, Hungary, Italy, Portugal, Slovakia, Spain, and Turkey. Well, as you know, as you start to understand, this is a global problem starting in the beginning of this century, maybe 2005, there are the year where the first news about this problem start to appear in the social network, in the newspapers, in the articles. Well, concerning the impact in each country. Concerning Austria, they first detected the Gnomoniopsis in 19. As Johan said me, in this year, the lack of wild chestnuts from the forest caused a very strong demand for fresh market, and all our producers were sold and quickly, and the fruits were soon consumed. That means that maybe the problem is not so increased, because one of the biggest problems is the post-harvested, where the fungus can increase a lot. Well, so at the harvest time, we recognized only very few rotten nuts between 0 and 50 and 5%. Concerning, but as Johan said me, well, if they storage the nuts, well, the rotten increased a lot for in 2-3 weeks for 25-30% of rotten fruits. Concerning Dryocosmos, first signs were detected in 13. Now the situation is controlled with thoriums naturally imported from Hungary and Slovenia, as you can see, because they are not allowed to spread thorium sinensis in Austria. Well, concerning France, Geraldine sent me this diapositive showing that, well, the problem was increased a lot, as you can see in the left to right, and from 22 to 23, as you can see, from 4% to 16%, as you can see here. And concerning yeah, okay. Concerning Georgia, the other, another answer I had, they said me that, well, at this moment, no scientifically confirming the presence of Gnomoniopsis is reported in Georgia. But until now, the Gnomoniopsis is not in Georgia. But they said me that, well, the most important problem they have in Georgia is about Crephonesia, blight disease. Also, they are no research or report about the presence of the Dryocosmos corypheus in Georgia. Concerning Greece, Stephanos send me this text you can watch here. Well, they have a problem, a big problem, as you can see. Look, in 22, only Malivia Corporate had 700 tons, which could not be sold, 700 tons in 22, as Stephens said. They organized a meeting with the government, asking the government to have some compensation, but unfortunately they didn't have positive answer about this. Concerning gall wasp, the first symptoms, the first detections in Greece were in 14, and they started to control, to spread thoriumus in 18. Concerning Hungary, well, first they first detected, the first year where they detected the Gnomoniopsis was in 23. They are suspecting that they imported the problem from the chestnut. They imported from Chile in very bad conditions, and they composting the chestnuts, and they think that it is the cause of the actually Gnomoniopsis, the presence of Gnomoniopsis in Hungary. And concerning Dryocosmos corypheus, yeah, they have, but the situation is controlled, as you can see. And one more time, the problem is Cryphonetria parasitica. Concerning Italy, I have very few information. I suspect that Guglielmo, after my presentation, will present in more detail some data about Italy. In Lazio, the impact was about 30–40%, depending the areas and varieties. As you go to see, the impact of disease is more or less connected with edaphoclimatic conditions also. In Piedmont, they said me that, well, maybe in 10–12, but it depends. As you know, the first galls in the Piedmont were detected in 2002. This is a problem that comes from the first years of this century, from the Oceania, New Zealand and Australia, and spread around all the world. And maybe Italy was the door of entrance of the Gnomoniopsis in Europe. Well, concerning Portugal, Alcino Pires and Alcino Nunes, they said me that, well, this was the worst year we had since the last years in what concerns to the bad nuts, to the impact of Gnomoniopsis. They said me also that the problem was very bigger, for the first cultivars, September, October, hybrids and Portuguese varieties, national varieties from Castanea sativa, where around 40–50% of nuts were rotten, and the cultivars harvested in the late of the season, Longal, the most important, had only about 15% of damage freeze. The question is, the problem and the resistance, the tolerance is from the cultivar or from the edaphoclimatic conditions? Well, in Portugal, this impact, if we estimate that around 30%, we can have a loss of 15,000 tons. That means about 30 million euros. Concerning Spain, in Galicia, they have a big problem since last years. 23 was one more year with the problem. Really, they start to think about Gnomoniopsis in 20, where a big problem with rotten nuts appear in the harvest. That means that 20 is the year of the first detection, officially first detection of Gnomoniopsis in Spain, in Galicia. Well, in Leon, they are doing some studies, and they found very different percentage depending the localities and the varieties, between 10 and 90% of impact in terms of varieties and localities. That means that in general the loss are about 32%. Interesting is that in Extremadura, in the middle of Spain, they aren't symptoms, they aren't losses due to the Gnomoniopsis. No Gnomoniopsis and no gallowasps until now. The biggest problem they have in Extremadura is the dryness was the dryness of the chestnuts. And a diapositive sent me from Spain, from Albert Sola. As you can see, the study he did about the Gnomoniopsis, where you can see in the right side the differences between varieties, changing between 10 and 16% of impact. Concerning Slovakia, until now, Gnomoniopsis is not a big issue, but maybe it is in nature, as Michel said me. Gall wasp was first detected in 14, and well, and he said me that, well, but we have gall wasp, but it is not a problem because we naturally imported also the thoriums to control the gall wasp. Concerning Turkey, the data are very scarce at this moment. First detections was in 21, maybe around 20%. Concerning gall wasp, the first symptoms were detected in 14, in Asian, Marmara, and Black Sea. And there are at least one variety that is comproved resistant to gall wasp, that is the Ayrton variety. And to finish, this diapositive to leave as a reflection. If we estimate the European production more or less 170,000 tons, that means if we have an impact of 30%, that means that we have a loss of 111,000 tons. That means that every year, if we repeat the years as 23, maybe that means a loss of every year of 222 million of euros. That means, and to finish, too much to forget this problem. Thank you.
Thank you, José. We have understood the state of the problem in Europe. I suggest before we move to the second part of the webinar, where we will ask the question of the factors that promote chestnut rot, we can ask the question because the question about this first part, there is 3 questions. The first one is do we know where the Gnomoniopsis is coming from? Do we have studies about the original place and environment of Gnomoniopsis? José, do you have something on this?
So if I understand, the question is about the origin of the Gnomoniopsis.
Where it's coming from?
Yes, the articles and the studies, the references said that the first symptoms, the first detections was in Australia, New Zealand. And after that, the Gnomoniopsis starts to spread around the world, from Oceania to Europe. And if I'm not mistaken, the first entrance in Europe was in Italy.
Geraldine,
yes, say, Andrea.
Can eventually add something about it. So I think that the main point is the confusion in taxonomy, because we know that what we call now Gnomoniopsis castanea or Gnomoniopsis middelvilgi was before Phomopsis castanea in a bona fide collection, because it was in a different fungal collection around the world. According to what we are studying now, because we did the complete genome sequence and now we are analyzing proveniences from, together with other colleagues, with Giorgia and other colleagues from Viviana and others that provided the proveniences, there is a huge biodiversity everywhere. So it's very difficult at the moment to assess if there is if it will be possible to find an origin. It seems that this fungus is an endophyte that move with the propagation material of chestnut. So where there is chestnut, there is apparently this fungus. I think that, and then the colleague from Piedmont that studied a lot the epidemiology of the fungus can support my feeling that there is a huge problem of climate in the outbreak of this problem right now, because according to me the fungus was present everywhere since a long time, even before these huge symptoms appear in the last 15 years.
It's difficult to think that just with propagation material we could have a so wide epidemic almost contemporarily all around the world.
Okay, and do you have information about the situation in Asia, China, Korea, and Japan? Do you think
they have different species? They have different species of Gnomoniopsis that were published in the last 5 years. At least 2 more species that apparently are associated with similar symptoms in Chinese and Japanese chestnut cultivation. I don't know if anybody has records of our species in Asia. As I know, this is what is known since now. Different species, but always associated with chestnut.
Yeah.
Okay. Okay, thank you. It's time to go to the second part of the webinar. So, what are the factors that promote chestnut rot? I would like to give. Yes,
Geraldine, in chat we have some more information, some more notes. For instance, Miguel Angel said that in Spain, in the Bierzo region, the amount of affected fruits is greater than 80%. 80%.
In the Bierzo.
Yeah. There is another question, maybe, about the pollination of longal. I saw something about this. The late cultivars in Portugal also have a late bloom. Yes, a little, not so much, because in terms of harvest, the difference between, for instance, Judia and Martainha and longal could be more or less 15 days. In terms of floration, 1 week, no more, between the difference. The question is that there are some phases after flowering that are more or less longer, depending on the cultivars.
Yes, but the question of variety is coming next.
Yeah, of course.
Yes. So Guglielmo Lione is here today. He works at the University of Torino, and he has studied the impact of climate on chestnut rot. Okay, Guglielmo, we can see your screen. That's perfect. I let you make your presentation now. Thank you.
Thank you very much, Geraldine, for your introduction. During this presentation, I would like to share with you the results that currently are known from scientists about the effects of climate and of climate change on the incidence and sporulation of this important pathogen of chestnut. Let's start with the very basics of plant pathology. We do know that to have an outbreak of a disease, we need a susceptible host, we need a pathogen infecting the host, but because the disease can show, we need a suitable environment. And in the case of Gnomoniopsis castaneae, we have different diseases on chestnut. We have nut rot, that is the most important one, but we have also foliar necrosis and cankers on shoot and branches. And the most relevant variables of the environment that can influence diseases are climatic variables. Moreover, Gnomoniopsis castaneae is a very complex fungus.
Guglielmo, sorry, but you are not sharing the display of your computer, the presentation. I'm sorry.
Really?
Apparently, yeah, I was talking on the first slide. Now, yes. But
Maybe can you put it on full screen, please?
Yeah, it was actually on full screen. I don't understand why. Now you can see it.
You have two displays, two ecrans in your table. Maybe you are.
Are you on two screen?
Yes.
Guglielmo. Guglielmo, are you using two screen?
Yeah, I'm using the browser. Yeah.
Because if you're using two screen, just one will be displayed in the.
Can you see it now?
Yeah, but always the same.
Now?
Yeah. Try to put in presentation mode.
It is
please.
already presentation mode.
No.
Not for us, but...
Stop the sharing. Stop the share.
Okay.
And start newly, please. Stop and start.
Okay, let's start again. Is it okay now?
Yes.
Yeah.
Oh, fantastic. So I was just saying that Gnomoniopsis castaneae is a very complex microorganism because it is a pathogen, but it's also an endophyte. It's also a saprobe. And we can guess that climate may help the switch from the endophytic phase to the pathogenic one. So this is why scientists gave a lot of attention to the relation between climate and Gnomoniopsis. Before speaking about this influence, we should give some word about the biological cycle of Gnomoniopsis castaneae. That is a fungal ascomycete that has both the sexual and the asexual form. From the sexual form, the teleomorph, the fruiting body perithecia develop on the spines of the birds and unrotten nuts, and these are the main substrate from which the fungus can release the infectious ascospores. There is also an asexual stage of Gnomoniopsis castaneae that forms its infectious inoculum at the level of the galls that are induced on chestnut by the Asian gall wasp, Dreiocosmus coryphilus, but the asexual form can also develop on bark cankers. Both the sexual and the asexual phases contribute to the infectious process. From the birds, from the litter, the ascospores are released and they arrive when the chestnut is flowering at blossoming time and infect through flowers, especially through the female flowers. And then the fungus develop within the nut, giving and producing the well-known symptoms that the other speakers talked about. But we can also have secondary infections throughout the season from the asexual phase. We should not be worried only on chestnut orchards because we have also coppices and hardwood forests of chestnut that may act as reservoir of inoculum. So the question is how climate and climate change may act on the biological cycle and on the infectious process. We should ask this with a few questions. And the first one is, which biological and epidemiological dynamics are or might be influenced by climate. I listed some with the red square about the things that we don't know at the state of the art, or don't not completely. The green one to stress points we already know. So the climate may influence the reproduction mode of the fungus. And this is very important because boosting the sexual or the asexual form, we could have more prolonged infections. Climate may act on the production and release of the infection inoculum, also in time and in the total amount of production. Climate may help or may not help the spatial spread of the inoculum. Spores are airborne, but conidia can be transmitted also by the mediation of precipitation effects. Climate could act on the germination of spores and conidia, but could also act at the host level on the phenology and hence on the susceptibility of the host. It could have the pathogen to help the colonization of the tissues and the onset of the disease and boost its incidence in pre-harvest and post-harvest. But climate could also act on the survival and overwintering of the fungus. And as you can see, most of these points are still not completely known. But what is climate? Climate is a quite complex ensemble of temperature, humidity, precipitation, such as rain, snow, hailstorms, precipitation that may also hurt chestnut, make a lesion and help the fungus to penetrate the host tissue. Then we have wind, solar radiations. But it's not only a matter of which is the variable, but also how long climate on the host, how much, how frequently, and how many times we have a given variable, such as temperature, over some critical threshold. So why it is difficult to study the effect of climate on Gnomoniopsis castaneae? Well, as researchers know, we cannot control climatic variables in the field. But it's also difficult to carry out experiments in controlled environment because this is a disease of adult chestnut. So we cannot simulate realistically disease dynamics in climatic chambers on greenhouses. And then it's difficult to disentangle the role of climate from the role of other factors such as the cultivar, the management practices, the tree age, and so on. So, which is the point of research to date? We have some studies about the role of climate on the disease incidence. And these are studies that are quite complex because we need not only a huge field effort, not only laboratory equipped and specialized with biological, microbiological, and molecular tools, but we need also a lot of mathematics to find the functions that can relate the climate to the incidence of the disease. Some minutes ago, we were wondering which is the impact in northwestern Italy, where the studies come from. When some years ago, we had a very variable impact from a minimum of 20% up to 93% of incidence. And we noticed that there was a strong side effect on the incidence of the disease. By measuring temperatures on a daily basis from January to October, just before the nut collection, and rainfall, we used a huge and complex mathematical structure with partial squares and other complex models to understand the deep relation between climate and Gnomoniopsis. And we arrived to fit predictive models that starting from climate can help us in predicting the incidence of the fungus. As this graph shows, there were different clusters of sites. The warmer site, those that have warmer average mean maximum and minimum temperatures of the months before harvesting, were those showing the highest incidence of the disease, while we did not find any significant correlation with rainfalls. Which is the role of climate change? Our simulations show that if climate change means rise in temperature, in the future we might expect increasing incidence of Gnomoniopsis castaneae, and so an increasing impact and economic losses. We also studied the effect of climate on the sporulation of the pathogen, on its ability to produce infectious inoculum by using spore traps coupled with qPCR and other molecular tools. And the bad news is that the Gnomoniopsis castaneae is able to sporulate approximately all over the year. It sporulates comparably in fall, summer, a bit more or less in spring, and there is only a drop during the winter. So we have a constant, almost constant production of infectious inoculum. We also studied the impact of the climate on the production of spores and hence on the risk of infection. And interestingly, we discovered that also for the production of infectious spores, increasing temperature boosts the capacity of the pathogen to sporulate. But we also detected that those sites where there is more wind, so the windy sites, are those which have the highest probability of detecting high sporulation levels. These are not the only effects of climate that are reported in the literature. Noteworthy, in Australia and New Zealand, researchers studied and arrived to the conclusion that the rainfall at blossoming, at flowering time, could foster local infections at floral level by ascospore of Gnomoniopsis castaneae. Other studies suggested that also drought may foster the incidence of nut rot, maybe because drought stresses the chestnut and could eventually help the switch from the endophytic to the pathogenic phase. Another study also reported that high temperatures and relative humidity could help Gnomoniopsis castaneae not as a nut rot agent, but as a canker agent. So in conclusion, what we can say about the role of climate and climate change? Well, Gnomoniopsis castaneae is correlated in its incidence, impact, and sporulation with climate. Higher temperature levels, increasing winds, and rainfalls at blossoming times are probably the most relevant climatic factor boosting and favoring Gnomoniopsis castaneae. And if climate change implies the rise of temperature, we can expect in the future an increasing impact of Gnomoniopsis castaneae. This is why it's important going on with research, going on with networking, that finding to study more and more this relevant pathogen. I invite you to read two reviews, also one that is very recent by Dobry and Campbell, about the state of the art of the knowledge on Gnomoniopsis castaneae, and also the literature cited therein that comes from an effort of all researchers over the world facing this important disease. And I wish to thank you for your attention and the EUROCASTANEA network for inviting me to give this talk.
Thank you so much, Guglielmo. I would like to ask you, what is the current status of your research? Are you continuing the research to confirm your result, or does your research is at the end?
As you can see, we have a lot of points that we need to elucidate. The most difficult part of the research is not doing the research, but is to get adequate funding to support research. This is the main issue. In any case, we are continuing to study Gnomoniopsis castaneae and trying to collect data, also to improve our predictive models.
Yes, that's important. And a question in the chat, what does a canker of Gnomoniopsis look like? Is it like a canker of Gryphonectria? It's the same look?
Well, morphologically, sometimes yes, but the question of canker and Gnomoniopsis is very complex. It's very complex because Gnomoniopsis is an endophyte. Canker is mostly related to Gryphonecta parasitica. So when you try to attempt in estimating which is the incidence in the field of cankers related to Gnomoniopsis, the fact that you can isolate or detect molecularly Gnomoniopsis on cankers does not mean that Gnomoniopsis is the primary cause of cankers, because they could have been cankers from Gryphonectria, and then Gnomoniopsis could have been arrived later or could already have been inside of the chestnut. Some practitioners said that at the beginning, especially on very young trees, on little branches, you have a sort of tissue prolapse, but we don't have studies that clearly confirm morphological differences at the symptoms level. If we speak about the signs of the pathogen, we can, of course, distinguish Gryphonectria from Gnomoniopsis castaneae, but of course, you need to have some experience in plant pathology. It's not something that you can perceive by sight so easily.
Thank you. There is some question about treatment, post-harvest treatment, and I would like to say to you that we will see that in the last part of the webinar.
Yeah, I think that Andrea Vannini, that is an expert in this field, will be more qualified than me to answer to the treatment part.
We will.
What I just want to add is that if we notice that there is a relation between temperature and the onset of the disease in the field, we might think that also when we transport, we move chestnut, we can have an effect of temperature during transportation. This is just a further input.
Okay, so now I will give the floor to Magalie Léon Chapoux. Magalie is an arboriculture consultant in France, and she will share with us her feedback on cultural practices in orchards in the southwestern France. And she will also present some study results from the experimental station INVENIO and the Ardèche Chamber of Agriculture. So, good afternoon, Magalie. We can hear you. Go. Hello. You can go. Thank you.
You can hear me?
Yes.
Okay. So I introduce you 4 studies about influence of farming methods on the presence of internal fruit rots. Chloris, arbo, INVENIO, and la Chambre d'Agriculture d'Ardèche took part in those studies. This study one in a study influence of farming methods and the presence of internal fruit rot. In 2020, 36 samples were collected in 17 orchards. Fruits were incubated during 10 days at 20 degrees. I noticed 3 kinds of rots: gray rot, brown rot, and chalky rot, and the presence of insect larvae. I also noticed some explanatory variables: orchard exposure, time of harvest, frequency of harvest, type of harvest, total or chestnut only, aeration of the orchard, organic or conventional forming, orchard age, density, fertilization, and irrigation. So 4 orchards emerged with low rate of rotten chestnuts. For all the explanatory variables, we can see that some of them seem to be more important. That is the case, for example, for the orchard exposure. In the east and southeast is unfavorable for the disease development. For the same variety, is there an impact on the decision on the order of harvesting plots? Second explanatory variable, the fact that the orchards is more open makes its chestnuts more rotten. You can see the orchards. And without a surprise, when more frequent harvested reduces the rate of rotten chestnut. Okay. For Bouche de Bétizac, it's relevant to vibrate the trees. It reduces number of harvests, and there is less risk of rot development. But it's possible for just for a few varieties, like Bouche de Bétizac. So we couldn't make a conclusion for the other explanatory variables. But if we consider all the orchards, statistical analysis shows that for the orchards that have all those parameters, like you can see, old orchards, less density, conventional farming with irrigation and fertilization, total harvested with many passes, there is less risk to develop disease. The second study is a comparison between the observations have been realized after cleaning the chestnuts and during comparison between organic and conventional farming. The observations have been released after cleaning the chestnuts and during 3 seasons. So we can see that there is no significant difference. Okay, the study 3. It's the impact of orchard thinning on the fruit rot development on Bouche de Bétizac. Orchard 1 tree out of 2, 1 row out of 2 were removed. It was progressive pruning. Orchard 2, its reference. there are no pruning. There is no pruning. Orchard free, major pruning intervention with pruning with a cutting bar and 1 row out of 2 removed. So here you can see photos about the orchard free with a cutting bar. The results. And the observation was made before cleaning chestnuts. And you can see that there is a tendency showing the positive impact of pruning on the percentage of rotten fruit. So it's different from that we have saw during the first study. And the study 4, it's the impact of the pruning of high density orchard. The modality is the restoration, of the tree by cutting back branches 1 tree out of 2, and after 5 years the same operation is cut back branches on the other trees. So the results, there is no significant difference between the two modalities. The observation were made from 2011 to 2021. And other parameters are noticed, donc with the temperature. The rate of rot is higher on years when summer is hot and June is raining. It's a conclusion of this study. So to conclude, these partial studies show that climate plays a major role in the infestation and development of chestnut rots. However, to reduce the damage, the grower can take action by prioritizing the east-southeast exposure of their plot when planting, if they have the choice, increasing the frequency of harvest on the most sensitive variety, variating trees of varieties that allow it, bouche de Bétisac, preferably carrying out a total harvest, chestnuts and bogs and birds, preferably fertilizing and irrigating their orchards. For the moment, the studies do not show a significant positive effect of organic rather than conventional farming methods, heavy pruning of trees, and tree density. Thank you.
Thank you, Magalie, for your presentation. Stay with us. Maybe there will be some question in the chat. The result seems to be seems to concord with Guglielmo studies about the climate. Now we come to the last part of the presentation, and not the least, because we are going to talk about the means of disease control. So what can be done to control attacks? That's the question we ask to the European teams. And we are going to see now maybe the first piece of good news, because they would be an effect of varieties. So this would be a lever for the ORCADs of tomorrow. On this subject, we will listen to Sébastien Cavaignac. Hello, Sébastien.
Hello.
Sébastien works at the chestnut producer experimentation station in France, and he will present the result of the variety responses.
Hello, everybody. So my presentation will be more on observation. I will try to make some picture of situations in order to show you some results of different varieties, cultivar responses. You already see in the first part that there are some piece of answer for this. First of all, I would like to thank all people who helped me to make this presentation by giving me different kinds of data. You find their name here, and they already have a presentation for some of them in this afternoon. So many thanks for your information. And the different data types that I will show you this afternoon are some information from experimental situations where we have different varieties in the same place. So it's very interesting because it's the same pressure and same climatic condition. We have a long time series and a strong protocol to follow the level of rotten fruits. We have also data coming from farmer sites. It's very interesting because we have a global information and another information in a different place. And in this place, we have more often less varieties follow in order to have this information. And the third kind of data I will show you is information coming from agricultural cooperative when we are bringing the nuts and giving us the aggregate results. So it's kind of interesting because we are on big volumes, but maybe less accurate than experimental or scientific situation. So the first image as introduction is the result of rotten fruit in cooperative. We have here near INVENIO in southwest of France. And what we can see at this level, it's a kind of confirmation of what have been said before. It's that before 2015, only 1 year was clearly under over 10% of defect, and from this year it's a kind of opposite. Now we have very few years with less than 10%. And what we can see, we have different cultivars, Bouche de Bétizac, Bournette, Marigold, Marsol, or Precoce Meagle. And what we can see is that some years, some cultivars are more rotten fruit than other years, and it's not always the same in function of the year. So maybe we need to go further in order to understand the different behaviors of the cultivar. So that's why we are making study quite more scientific when we are on the site with the same condition, and we will collect fruits, and we will hear the results I am showing. It's a kind of harvest information. It's not with incubation like present Magalie before. It's harvesting 1 month at -2 degrees and looking the percentage of rotten fruits. And what we can see in these 4 years, it seems that some varieties seems to be less sensitive. At the left, sorry, of the picture, Jeannette, Belfer, Bournette, Verdal seems less sensitive, and at the right we have some which seem very more sensitive to Gnomoniopsis. And what is difficult in this analysis is that some years there are some unexpected behavior. You can see that, for example, Montaigne in 2020 was very few problem of rotten fruits, and for example, Bétizac in 2022 has a different behavior. So there is still some question why we have this kind of heterogeneity. But if we take a scope out, we can see that we can class between less sensitive and more sensitive varieties. This study was also made in other parts of France. You have here some on the left some results in southeast of France, where we can find, if we are looking at Bouches de Bétizac and Belfer, this kind of classification, and we can find two where you have the arrow, the pink arrow, this difference of behavior which seems not expected. And on the another results coming from CTIFL, we can also have this difference of sensitivity between varieties, where we can find the same kind of classification between here Bouches de Bétizac, more sensitive, and Belfer. So we can have this kind of classification. But if we look at a second observation, which is after incubation or after a few days of storage, the comportment will be quite different. Sometimes after 20 days at 10 degrees, you can have more a kind of explosion of rotten fruits inside inside the nuts. And what we can see some years, a kind of inversion. If you see, for example, between Belfer and Bouches de Bétizac in 2023, there were less defect in Bouches de Bétizac than in Belfer. But if you are looking on a long period, we can most often find the same sensitivity between the results at the orchards harvest and after conservation. And there are some cultivars like Jeannette who present every year good results. So we can see that cultivar can be an answer. Here we have some image of varieties after the test, when we have more than 80% we are non-commercial marketable. But on the same test, when we have the same treatment on Jeannette and Belfer, the percentage of marketable fruits are less. So we have a good way first way to answer this problem. Some other results coming from other country. You have on the left a result which I've already shown this afternoon, where we can find this difference of sensitivity between different cultivars. So it confirms that there can be difference. And you have on the right a result which is quite interesting, because it's made from inoculation, artificial inoculation into the nuts. And doing that, we can see that there is a difference of response. You have to see the two clear blue at the bottom to see the difference of response between the different cultivars. So we seem to be able to class very sensitive, like Bouches de Bétizac, which is also sensitive, famous in Australia, I see in the first part, and less sensitive. And we try to look if there is a link between the harvest period and the sensitivity. Links doesn't seem so clear. We can find sensitive cultivar who can harvest early, late, or medium, but in less sensitive, there is not really a late harvesting. So maybe there is something to start to understand the link between the sensitivity and the time of harvest. It's just a new way maybe to understand. And in conclusion, what I would say is that there is no binary answer like a variety which is resistant, and there is no problem of Gnomoniopsis. There is no this kind of binary answer. But when we are looking on long time series and comparison between cysts, we can see that we can give a level of sensitivity between different varieties. So if we want to manage the risk, we can here have a choice for future orchids, like was saying Geraldine. There is still some unexpected result, like I show you, that it means that we still have to make study and to understand why there is this kind of behavior in order to bring a solution. And what we can see that at this sort of art and of information, we have to find to fight Gnomoniopsis at all levels: prophylaxis, during culture, harvest, post-harvest, and it's in a kind of introduction to the following presentation. Thank you, everyone.
Thank you very much, Sébastien. So now we can continue the presentation on the theme of control with the presentation of the results of the research in the United States. So Giorgia, can you make the end of your presentation about your results? Thank you.
Okay. Can you see it? Can you hear me? Hello? Can you hear me?
Yeah.
Okay.
Okay.
You can go on.
Okay. So again, it's me, Giorgia Bastianelli from Michigan State University. So after what have been said for the first objective of the project here in Michigan, some control trials have been conducted at 2 colossal orchards located into a different research station in Michigan. And colossal, as we saw before, is actually the most susceptible cultivar to brown rot here in Michigan if compared to other cultivars. And for this reason, trees has been sprayed in the second half of June, when female flowers were receptive for pollen and so also for spores, and then after 2 weeks when the bud was shaping. And these are the chemicals that have been used last year in June and July. And most of them belong to group 3 fungicides, the azole group. And azoles were proposed since their efficacy was already demonstrated in Italy and Australia in some published papers. And in this case, we are talking about tebuconazole and difenoconazole. And then other chemicals are used and were used and evaluated for the first time to the best of our knowledge, like propiconazole, always group 3 fungicides, and azoxystrobin, group 11 fungicides. And we also included a treatment with potassium phosphide, a systemic defense inducer for plants, since it was used earlier in Italy. And this approach should provide growers sufficient fungicide treatments to rotate application during the season to reduce the risk of inducing resistance of Gnomoniopsis or other fungal species. But before testing the different chemicals in field, we tested the fungicidal activity in vitro conditions, so in laboratory, and with petri dishes amended with chemicals in different concentration. And the result in AC50, the dose that provides 50% of inhibition of the isolate as compared to an untreated control, were similar for all the chemical tested except for K5, so potassium phosphide, which showed a significantly higher EC50. But this was expected if we think that potassium phosphide has a very low fungicidal activity, is just an inducer of resistance for plants. So it could be identified as a fertilizer, not as fungicides. Anyway, between the regular fungicides in Spice Super, so difenoconazole, and then Tilt propiconazole, resulted the most effective fungicides in vitro condition. And these are just preliminary results on brown rot incidence in the 2 orchards we sprayed last season. And on the graph for the 2 orchards, you have the incidence right after harvest, and then after 1 month in cold room. So T30 means 30 days of storage in cold room. And of course, all the treatments had significantly higher incidence after storage than right after harvest. And it's interesting seeing that potassium phosphide was the only treatment to statistically differ from the control after 1 month of storage in the cold room, while no one of the other treatments differed from the control from the 2 times of storage considered. And concerning the severity of the disease, no statistically differences were found between the treatments and the control, considering both nuts right after harvest and after 1 month in a cold room. These are just preliminary results. So the next steps provide for qPCR assays on the DNAs extracted from the chestnut from last season in 2023. And we are right now running these qPCR assays using the protocol developed in Italy by Silvia Turco in 2021. And then the next steps provide also field treatments using the same chemicals of 2023, plus some other chemicals that I know that has been used in Italy and are really promising, has some zinc-based biofungicides or fertilizer and zinc phosphide fertilizer. And this is going to be done in June and July, and then all over the summer, increasing also the number of treatments during the season. Then for sure, before testing these chemicals, these new products, let's say, in field treatments, we're going to evaluate the AC50 also for these new products during the spring. And then we will test the efficacy of post-harvest treatments in Michigan during the fall, so at harvest, in the harvest period, since we saw before that some steps during the chain discard an excessive high number of healthy nuts that are wrongly identified as contaminated by brown rot or other fungal pathogen. So thanks you all for the possibility to share this data in this webinar.
Thank you. Giorgia, there is questions typically for you.
Yes.
What temperature were the cool room in the trial?
It was between 0 and 4 degrees Celsius degrees.
Okay. And the second question is, what is the application methodology used in chemical application?
Just crown spray. We didn't use any other application. We are planning to do some trunk injection as we did earlier in Italy, but for now we just did crown spray applications.
And when? Which phenology of tree?
When the flowers were receptive for the female flower were receptive for the spores. So basically when the flower it's wide open and receptive for pollen and spores. Okay. It depends on the season, but usually it happens at like in middle June or the 3rd week of June.
Okay, and is it possible to have the number of application maybe in another?
Yes, we just did 2 applications this season, one in June and then one when the flowers were in this particular phenological stage, and then one after 2 weeks when the birds was like appearing for the first time, was shaping. But next season probably will increase the number of treatments also during July and August. But we are still working on the next season.
Okay, the study is you continue the research.
Yeah.
Okay.
Yeah. Also, not just increasing the number of sprays during the season, but also increasing the number of chemicals that we would like to test.
Okay, thank you very much, Giorgia. Now it's time to listen to the last presentation. I welcome Professor Vannini, who works at the University of Tuscia in Italy. Professor Vannini will present the main result of European research on chestnut rot, both on orchard treatments and on post-harvest strategy. So, Andrea, we listen to you.
Can you see the presentation?
Yes, we can.
Okay. Thank you very much. I'm very happy to share this presentation with so many people. I'm very excited that thank you, Geraldine, for the work you have been done. Okay, let's in this presentation I will try to summarize, if I can go. Okay. The results that were I don't know. Okay, sorry. The result about control trials that have been done in a different country in Europe. I collected the information from France, Portugal. I have some information, of course, from Italy. And we will summarize these results and see which is the state of the art. Okay, I will skip these. You know a lot about Gnomoniopsis at this point of the seminar, so I can also skip symptoms and so on. Just to say that to answer about the question of from where Gnomoniopsis is coming, based on the genome sequence of the fungus, we could develop microsatellite that we are using now to screen the world population of the pathogen to see if we can understand something about the origin. I don't think we will arrive to a clear result because of what we have been saying before about the ecology and biology of this fungus that is basically an endophyte that move with the chestnut. Okay, this is the present. This is the more or less the chestnut production in the world, and this is the chestnut, the presence of Gnomoniopsis. As you can see, is present everywhere. The chestnut is cultivated at the moment. Okay, this is, for instance, is what happened in Chile. Viviana already sent show us the results. This is the chestnut that arrived in Italy in April 2023 from Chile. It was the first year that we noticed this such heavy infection of fruits from Chile. I just want to recall you that in Chile mostly Castanea sativa is cultivated, varieties of Castanea sativa, marrone, and other varieties. Okay, what we are going to show you is based on what Guglielmo and before the group of Guglielmo, Turin, and before the group of Shuttleworth did about epidemiology of this fungus. Most of our treatment were based on phenology of tree, as Giorgia told before. Since has been demonstrated that the pathogen penetrate the female flower during blooming, the most important treatment is the treatment done during blooming, and this is what the different group did in their experimentation. Of course, as Giorgia and others showed before, fungicide control against Gnomoniopsis is working with tebuconazole, propiconazole, and other product. But we have some problem with this product. First of all, tebuconazole is going out of the market in Europe. There is a derogation until 2026, but then it will be banned from application. And the other point is that in Europe, many chestnut cultivation areas are basically agroforestry, and we don't want to use chemicals in a forest-like environment. So let's start with the experimental trials that has been done in France. Sébastien sent me the data about 15 days ago, and I tried to summarize the data of Sébastien that are quite interesting. They did trials using a wide range of product, spanning from phosphonate, potassium and zinc phosphonate, microorganism like Gliocladium and Bacillus subtilis, algae extract like seaweed Clonia maxima, and then of course fungicide, chemical fungicide like boscalid, pyrasclotrobin, fluxonil, and tebuconazole. They repeat the trial for not for all the product, but up to 4 years. And the results are quite interesting. I will summarize later on all the whole result of this experimental done in Europe, but basically there are a solid, a consistent efficacy of Bacillus subtilis in reducing the rate of infection in fruits. In this case, there have been done three treatments, one in blooming, one at pericarp coloration, and the other at the end of chestnut of the fruit grow. Of course, there are good results about tebuconazole, but we know that tebuconazole is working, so it's the point is how to substitute tebuconazole with other less impacting product, and for sure Bacillus subtilis is one possible alternative, because there are several commercial product on the market based on these bacteria. Very interesting, even the results from algae extract, but unfortunately there is just one replicate, one season done, and it would be necessary at least to have a couple of seasons of trials in order to have more solid results. Okay, let's see what has been done in Portugal now. In Portugal, they did several trials in the different years. This is the last one done in 2023. They also use potassium phosphonate. Then they use a microorganism, Gliocladium catenolatum, very similar to Gliocladium, to the experiment that to the trial that had been done in France. And they use also Bacillus moyavensis, that is different from Bacillus subtilis, but still present on the market as a EM of phytoagron. Okay, the results, of course, are divided in developing isolation of Gnomoniopsis and other microorganisms. There is as a good result from potassium phosphonate, Gliocladium, not very good results with the Bacillus in respect to the control. We don't have the statistic of this, so I cannot say if differences are statistically or not. After 30 days of storage of the fruits at 4 degrees, just pre-stop, that in this case is Gliocladium, keep the infection half compared to with the control. They did another experiment using fungicide like using Bacillus subtilis, and the market product is Serenade in this case, tebuconazole, and high concentrated silicate. This experiment was done in 2021. In this case, also, as expected, there was a good efficacy of serenade, that is Bacillus subtilis, that is the same results more or less that the French team obtained. Of course, there is a good result for tebuconazole, but we know that tebuconazole is working. The last one was with zincophosphonate. That is another sort of phosphonate that we have been used in Italy for 3 years in trials, and compared with the tebuconazole that has been done in 2022. In this case, it was demonstrated the efficacy of zinc phosphonate to reduce the infection at the time of to reduce the impact at the time of harvest, but even good results after 30 days of storage at 4 degrees. Okay, let's see what we have been done in Italy. We start in 2019 with different product. We replicate in 2020 and 2021. 2022 and 2023, using basically as a positive control the tebuconazole, and the market product is Mystic, and then use a different phosphonate salt that was the potassium phosphonate that is Calyx, a zincophosphonate that is Calyx ZN. And this is the experimental design. It was completely randomized in 2020 and 2019. How did we treat? Most of the treatment have been done with the crown spray, as you can see from the left. Treatment with the potassium phosphonate has been done through endotherapy. Those are the results based on different indicators. The first indicator is isolation of Gnomoniopsis from fluids. As you can see, in 2019, potassium phosphonate, zincophosphonate work as well as the tebuconazole compared with the control. Similar results have been obtained with different indicators, with incidence and severity in 2020. While the potassium phosphonate didn't work that so well, but it is probably due to the fact that the time of injection were different, and probably we didn't obtain the translocation of the product to the flowers on time. But zincophosphonate works very well through crown spray, with one treatment at blooming, or 2 treatments, one in blooming and one in full August, in the time of ripening of the fruits. And this is another indicator based on qPCR. So we test molecularly the presence of the fungus into the fruits. And even in this case, the effect of Calyx zinc phosphonate was very good, comparable with the effect of tebuconazole. Okay, but this is the residues in fluids of phosphonate, zinc phosphonate and potassium phosphonate is largely below the threshold according to the European regulations. But we have, and these are the SC50. I fully agree with what George has told before, that it is extremely important to conduct the C50 in the laboratory, because this will avoid misunderstanding in field. Because in field, according to the season, some product can have a better effect than in other season. But if we know that the product is working in the lab with the C50 against the pathogen, we can better interpret the results even in the field. So my suggestion for everybody is conducting trials in open field is extremely important, even to conduct in the laboratory the test of the efficacy of the product against the pathogen. Which is the problem? The problem is that we were very happy that zinc phosphonate was working, but unfortunately the phosphonate has been banned from Europe as a fertilizer. So now must be registered as a fungicide to be used, and of course must be registered on the culture, on the tree. And at the moment we have nothing registered on chestnut. Just recently in July we got the registration of Century SL on chestnut, that is potassium phosphonate, but about zinc phosphonate we have no hope to have the registration of this product. So we need a new eco-friendly solution. In 2023, we conduct new trials using different product, having as a comparison the zinc phosphonate. We were using another fertilizer that is the Calyx Evo, and the Chanoil plus Target, and the Fungicrops, that are all products that can be used in organic farming. Okay, the results were quite encouraging. We knew that zinc phosphonate is working, but was very interesting that Fungicrops Bio was working very well. And even the Chanoil plus Target that was working. Calyx Evo was not working that good in the trial that we have been done. Fungicrops Bio can be used in organic farming, of course, even Chanoil and Target Plus. Okay, this is Fungicrops. It's produced by LIA Ltd, that is an Italian farm. It's a mix of manganese, molybdenum, and zinc. This is very interesting because zinc is coming again, being also present in zinc phosphonate. And this is a Chanoil Zeta and Target that is from another company that is Agrigest, that even combined together, they are extract of algae. And this is extremely interesting because the French team obtained very good results with the algae extract as well. And so there is some overlapping with the results of the French team. Unfortunately, this is just the first trial. We have to repeat the trial this year in order to have more solid and publishable and transferable results. Okay, this is to summarize what happened in Europe about treatment. I summarize all the product that have been used in the trial in French, Portugal, and Italy. And I just want to highlight that two products demonstrate to be being replicated in trials, and different trials in different years, demonstrated to be very efficient. Bacillus subtilis with the market product Serenade and Rhapsody, and zinc phosphonate with the Calyx ZN. Unfortunately, as I told you, zinc phosphonate cannot be used in Europe anymore, but can be used in the Americas, and can be used in Australia, probably can be used in other parts of the world. good results has been obtained with the algae extract, but unfortunately there is just one trial. I will encourage Sébastien to repeat this trial in order to confirm the results. And even with this mix, and both good results have been obtained even with this chanoil and target. It is a mix of algae and vegetable extract, but even in this case we just have one trial. Again, very good results have been obtained with Fungicrops Bio, that is a microelement combination. Even in this case, we have to repeat the trial next year. Tebuconazole is working, but we hope not to be forced anymore to use this kind of product in our chestnut plantation, especially in agroforestry system. However, I want to stress the point that the data that we have been showed are not comparable each other because there are differences in timing, doses, way of treatment. So probably in order to have comparable results, we need a more coordinated activity in trials. And there is a huge problem that was already highlighted by different colleagues, that efficacy of treatment after long storage of fruits represent a problem without post-harvest treatment. What is the problem? The problem is that the bucanazole is a fungicide, but the other products are not fungicide. They are basically fungistatic. They reduce the rate of infection, but the presence of the pathogen in the fruit is still is still there. So without a good treatment in post-harvest, it will be difficult to solve the problem. Let me summarize what we did in post-harvest in the last 2 years. This is we applied. We were studying the different phases of processing of the fruits in a modern plant, and the business as usual situation is very dramatic, because Gnomoniopsis is present in all the phases, and the presence in storage is commonly increasing. There is a nice indication that after sterilization, apparently the rate of infection decline, but then those sterilized fruits in storage develop the symptoms again. So there is something wrong with the processing. We were studying the processing. We find out a threshold of temperature that were able to completely inactivate the fungus in post-harvest. The treatment is at 50 degrees for a minimum of 30 minutes. Depending on size, I should say depending on size and state of the infection. But basically, a treatment of 50 degrees for 45 minutes is a very, can be the treatment that completely inactivates Gnomoniopsis in post-harvest. This is a very interesting, because even another group in Emilia-Romagna did the same experiment, obtaining the same result that we were showing before. However, there is a different ability of temperature to inactivate the Gnomoniopsis in fruits, depending on the severity of the disease in fruits. Higher is the severity of the fruits of the disease in fruits, longer should be the time of sterilization. Anyway, we found out that we can keep for a maximum of 45 minutes. Why? Because after 45 minutes, the organoleptic characteristic of the fluid are starting to be lost. While if we keep a sterilization phase of 30 or 45 minutes, we have no difference in the organoleptic quality of the fluids according to a blind test that we did with the consumers. Which is the problem? What you have seen up in the left is a sterilization plant. The sterilization plant has a panel of control where you can set the temperature and time of sterilization. The problem is that the mass of chestnut that entered the sterilization entered at a different temperature because came from floating phase and commonly are cold and keep and take a while to reach the real 50 degree. And this is what we have to assure that it will be an effective sterilization at 50 degrees for 45 minutes. What we are going to, and this is the problem in upscaling when we move from the laboratory to the plant. What we are going to do this year, what you are seeing in the right down is a micro sensor of temperature that we will put in chestnut in order to monitoring during the sterilization phase if in the reality the chestnut will reach the right temperature and how long they will reach the right temperature. We will start this experimentation in the plant this year in order even to give information to the manager of the plant how to set the machineries into the plant. Okay, this is what we are going to do in 2024. Let me just go very fast on another problem that is the problem of mycotoxin producing fungi. What I'm talking about the post-harvest method of sterilization are remediate the problem of Gnomoniopsis, but do not remediate the problem of fungi, of mycotoxin producing fungi. Fortunately, I will be very fast here. Fortunately, we were studying, while Giorgia was my PhD in Viterbo, we were studying a different mycotoxin in chestnut during the processing, ochratoxin, fumosin, aflatoxin, and we found that the threshold, the presence of the mycotoxin are still below the threshold of European threshold, except for the mycotoxin producing from Fusarium, because we are very close to the threshold of acceptation of Europe. And we were studying the correlation between the presence of Fusarium and presence of mycotoxin, finding a very interesting correlation with some taxa of Fusarium present in fluids. Just a screening of the literature on other method of for instance, of storage using ozone, liquid or gas ozone. The results seems to be quite good for other contaminating microorganisms, but not very good for Gnomoniopsis. This is a very recent paper that say that even Gnomoniopsis is producing mycotoxin. At the moment, we don't know very much about the importance of this mycotoxin in the processing of chestnut fruits. So the upscaling in technology is very important. What I'm showing here is a new machinery that has been mounted recently in a plant we are working with, that is an automatic machine with 8 distribution line that can discriminate automatically up to 7 different defects of the fluids, 6 tons per hour, but unfortunately can discriminate only external defects. we were working in the last 2-3 years in diagnostic, non-destructive diagnostic of internal damage caused by Gnomoniopsis. This is a prototype that we developed in the last 2 years. But anyway, we were able to find a method to discriminate the internal damage. We are still at the laboratory test, but if we could integrate this method with this kind of machine, we are going even to have a very effective sorting at the end of the processing line. Okay. The conclusive remark is what has been told even before. We have to work in a complex system. Is not the efficiency of the field treatment alone is not enough. The efficiency of the post-harvest treatment alone is not enough. What we have to combine together to have a sustainability in the quality production of chestnut is the field treatment and the post-harvest treatment. That means that all the chestnut area should be equipped not only for a good protocol of treatment in open field, but even for a good plant for post-processing of the fruits. Next step, we will go on with the trials in 2024, new tools for early warning, smart monitoring of post-harvest process with the sensor I showed you before, and then they increase the technological readiness level of non-destructive damage detection tools. Thank you very much for your attention. This is our group. Our team working on chestnut. Giorgia is still here in this picture, and then she moved to Michigan. And this is the project that supported us in our activity. And thank you for your attention.
Well, thank you very much for the work of synthesis you made from all the teams work on the subject. That is very important for us to have this synthesis work. Thank you very much. So we are coming to the end of the presentation. Now we can proceed to questions and maybe open discussion. Maybe José and Bertrand, you can maybe, does Bertrand is here?
Yes.
So we can, maybe I can ask the first question. I would like to ask the question of tebuconazole to maybe Mr. Bourgoin is here with us from the Direction of Agriculture in France, and I would like to have his point of view about the status of this product. Can you hear us, Mr. Bourgoin? Ah, he left, unfortunately. Yes, I see in the participant list, and he just left. Okay.
José, maybe you know this problem. You know that, José.
Mm.
Do you have an answer for tebuconazole?
Well, the answer I have for Portugal is that, well, they said me that, well, hello, there is a derogation of tebuconazole for 26, and if you want to apply, but I have to ask newly, we allow it to apply the tebuconazole, but only during the pollination. is that they said me, is what they said me in Portugal. The question is that the application of tebuconazole during pollination induces limitations on the efficiency of pollinations. The trial we did 2 years ago, and I think that Andrea and Guglielmo has the same opinion, limited the efficiency and well, at the end, jogging, we don't have Gnomoniopsis, but we don't have also nuts, because it limits a lot the pollination, the efficiency of pollination. Maybe the possibility to apply tebuconazole immediately after pollination maybe could be a solution. But, well, the question is that tebuconazole go out after 26. Well, to solve the question, the problem we have this year and the next, maybe we could work and we could apply to use tebuconazole, but we need future solutions. This is the way of research did by Andrea, by Guglielmo, by my colleagues here in Portugal, Paula and Ana Sampaio, because we need another solution. Andrea talked about the products from bio with the good results. Maybe we have we are lucky. We have a good door for coming times. The phyto, I don't remember the name, the product, but a bio product with very good results also.
Yeah, José,
Yeah.
This is true, but again, of course, chestnut is not the same everywhere. We have a chestnut plantation in Portugal, in North Italy, and in France, that absolutely not comparable with what we have in Central Italy, that is mostly orchard that are
Agro-system, agroforest. Yeah.
We cannot use fungicide in forest. So
Yes,
our target is
Yeah.
to remove completely from forest this kind of product. Now, the product that we tested, again, are fungistatic. They are not fungicide. That means that we remediate to the infection, flower infection, but not at 100%. Without a post-harvest conditioning at sterilization and so on, the problem will not be solved. So what I recommend is that if you want to use alternative to classical fungicide like tebuconazole, you need a line, a processing line in the field, and in post-harvest, they guarantee even the sterilization and the correct storage of the product. If not, you have to use a fungicide. So you have to use something that is killing the fungus at time of the infection. But again, our results demonstrated that even with the tebuconazole, we are not going to solve completely the problem. Still, a fraction of the pathogen remain in fruits. Probably with this summer treatment, you can again increase the success of the treatment to going treatment in blooming and another in August, since tebuconazole is a systemic product. But then you look at the label of tebuconazole. I mean, tebuconazole is harmful for animal, is harmful for the water, is harmful for everything. I mean, at the end, we have even to pose the question to ourselves about the future of chestnut.
Yes, that's the problem. Yeah, but yes, we have a door opening is with the bio. You said, maybe it's a good opportunity because now we need to think hard to think for this year. What can we do together in Europe for this year in order to decrease the impact? Well, in some orchards, in some situations, using tebuconazole, yes. In another situations where we have bio, maybe the bio product you tested, well, it starts to give us some nice results. And well, tebuconazole is a fungicide, the other one is a bio product, and the complement of this application in the orchard that help the producers of in the bio with the treatment with hot water, maybe could be another way to help us. The other thing is that until now, we know that 48 degrees, 45 minutes were enough to stop the Tortrix deciduous. No, well, we need to increase 2 more degrees. 2 more degrees. Do you know if 2 more degrees is damage for the fruits or not? The fruits still stay good in a good.
At 50 degrees for 45 minutes, up to 45 minutes, there are no differences in blind test with the consumer on the organoleptic quality of the fruits. So
Okay.
there is no effect. The point is not this. The point is to have the equipment. The equipment is very costly. And the point is if to let the equipment work properly. Because this year I was talking with the manager of the plant, and I stressed the point. You have to keep the fruits at least 45 minutes effectively at 50 degrees. And they told me, we cannot do it because it's too much. Then we lose the timing of processing of the fruits, so on and so on. At this point, I told them, okay, so is no sense that you use sterilization. Turn off the sterilizator, you save energy, you save money, and you save time. I mean, because if you want to use the sterilization, you have to use the sterilization in the proper way. Otherwise, you are just wasting temperature, energy, and money and time. Sorry if I.
No, yes, Andrea, I'm thinking.
Bertrand wanted to say something, I think.
Yes,
I'm sorry.
Two things, several things. First, in France, I don't know everywhere in Europe, we have 60% of our orchards, or they are organic orchards. And we don't have a way with chemical fungicide. Is the first thing. Second thing, orchards, mainly orchards, are in mountains, and it's impossible to mechanize, to spray with a tractor and machinery. Well, the problem is very, very, very important. I don't know if we have a lot of time, Geraldine, for this discussion. We have how many? 10 minutes? 15 minutes?
Yes, all you want.
Okay, it's not invited. Okay.
No,
well,
because the end is at 5 o'clock and it's 5:30. So
okay,
now
okay.
the subject is too important. We can take the time.
The problem is very, very, very important. It's very important for producers, for growers. It's very important for fresh market. It's impossible to sell fruits on store with 20, 50, 70% of bad fruit. It's impossible. The market died in 1-2 more years. Everything is died. And it's the same problem for chestnut processing. If you have chestnut with 50 or 60% of bad fruits at the end of the chestnut processing, it's impossible to do that. It's a very, very high problem. Maybe it's highest problem than CNIPS. It's maybe the most important problem since maybe 1 century. Well,
Yeah.
And
Yeah,
I listen since 2 hours, a lot of questions, a lot of tries, but no very no answer very very evident. How can we do now? In fact, I don't know, but what I know is if everybody working on this problem work together, share their result, their tries, maybe we can find some solution. Maybe not this year or next year, but maybe in 2, 3, or 4 years. And we need that quickly, yes, but we must to work together. That we can do in Eurocastanea. Maybe it's to have a meeting like that 1 time a year with a lot of people to hear the different results. But in fact, that we need, it's a task force on this problem with all researchers, technicians working in this problem in the world. I don't know if we can do that with Eurocastanea, maybe, but we need a leader, maybe Andrea or José, and we need a group of maybe 50 or 20 or 25%, but a group very implicated in this problem, working together, sharing our result, our different tries. And I think it's important to start now, to start for this year, because June it's in 3 or 4 months, and we must to start early. And it's important to have people's researchers from Australia, from Chile, from United States, with all researchers from Europe. José, I ask you the question because you was the president for Eurocastanea before me. Are you agree to be the leader of a task force like that to continue the work on this problem?
Yeah. Thanks, Bertrand. Yes, of course, I agree. I'm honored to lead this task force. Only to remember that, well, we start to do something together. We here in Portugal, with Italy and with France, some groups of France, in order to start to work in order to find some solutions, because we need immediate solutions for this problem. As you said, is a big problem. After Galoisp, well, a new problem comes. And if, in what concerns Galoisp, the biological control works, and we need to spread insects at this moment, and I am afraid to say you that, well, to treat the trees, we need to spray. This is the problem. Even a fungicide or a biological product, we need to spray to spray the trees. This is a problem for the forest ecosystems. Contrarily to the Torino-Chinese, this is a problem. But anyway, well, we have is better than anything for these times. We have a biological product. Now we know that if we apply this product plus the treatment with water, we can partially reduce the problem. I'm thinking in the case of Portugal, we have a capacity to treat more or less many thousands of tons, because we have our exporters, our processing, our companies most of them have the hot water system. And in order to add what Andrea said, we had a big problem with Tortrix in 18. Brazil blocked the Portuguese exportations for Brazil due to Tortrix. What we did was in the hot systems, in the tank of hot water, they put 2 sensors, one in the beginning of the system and another one in the end of the system of the tank, in order to check the temperature of water, and these systems are calibrated in order to have sure that the water inside the tank is at 48 degrees. What they have to do now is to increase the temperature for 50. But our tanks are 2 sensors, one in the beginning and another one in the end.
Okay.
Okay,
May I say something?
Andrea have the hand up.
Yeah.
Andrea, and maybe the conclusion words.
But
May I?
yes,
Yeah.
Andrea.
I will be more optimistic of Bertrand, in the sense that
Thank you, Andrea.
that at the moment we have at least 3 products that work since that have been done trials for different years, and they demonstrate to work very well. That is Bacillus subtilis in the in the different marketable product. Okay, is a zinc phosphonate that still having the stock can be used until the stock are finished, and there is this fungicrops that is incredibly active, and even this different algae extract. Now, what we have to do, we know that they are working, and if coupled with the appropriate post-harvest treatment in hot water, they are solving, I don't say solving the problem because I'm not so, I mean, they are reducing sensibly the problem, okay? We still have some question points that are related to epidemiology, and this is referred specifically to what Guglielmo was saying. Because, for instance, this year we have a very strange situation in different parts of Europe, that the temperature were so high that chestnut stopped, the physiology of chestnut stopped in end of August, beginning of September, because you know the chestnut with temperature over 30, 32 stop physiology. That means that we have the chestnut, the fruits, ripening, stocking on the tree at high temperature. And that was dramatic for the developing of the disease, okay? So we still have this question point related to climate change. And basically we have to understand what we can do in case we have a similar season again. But at the moment, I think that treatment done, blooming, 1 week after blooming, and eventually in August, with the product I told you before, even in combination, I'm sure that they will reduce sensibly the problem on the tree. Then after, we should follow the rules. 50 effective 50 degree for 45 minutes, but should be effective. José is having the temperature at the starting ending point. We will put the sensor into the system, but we have to be sure that the equipment will be set having effectively 50 degree for 45 minutes. Otherwise, if this is 48 degree, it's not working. Okay, this is the point. If all of us will act like this, I think that we are starting to see the results since the next season. I'm completely open to share with the other group, Sébastien, José, Giorgia, everybody's working in control of Gnomoniopsis, to share the protocol, to use the same product together in the same way. This is the point. If we have to say that we have to treat when the flower is receptive to pollen, all of us has to treat when the flower is receptive to pollen. Otherwise, we have no comparable results. But in any case, I think that we are on the right way. We have, I mean, 3 years of trials done in France, in Portugal, in Italy, and at least 2 or 3 products that can be used in organic farming can be demonstrated to be highly efficient in controlling the disease. Okay, so I will not be that pessimistic, Bertrand.
Okay. Thank you, Andrea.
Andrea,
Yeah.
Do you believe to another technical instead of hot water?
Look, I think that hot water cannot be avoided, but might be combined with another treatment that some plants are doing upon our suggestion. That is ozone in water. But this is not much for the Gnomoniopsis, but for external contamination of other mold, and eventually ozone in storage room. If I can do it, I can just show you the processing. Can you see the?
It's very little.
Okay, now you can see.
Yes.
Okay. Basically, these are the different phases of processing. Coming to the farm, to the plant, there is the first screening, okay? And then go in, if necessary, go in cold water, but now just to resume the right level of humidity of the fluids, because sometimes the fluids arrive to the plant too dry, okay? Then go in the floating sink, where we eliminate all the fluids with the insect in the floating phase. That is the number 3. Then goes in sterilization. After sterilization, goes in another sink to cooling, to go from 50 degrees again back to 20 degrees, okay, of the fluids. And this is the phase where we use ozone in water. After we go to dry the fluids at pressure, hot steam of air. Then it goes in calibration, and then goes in storage room with ozone at 0 degrees. This is the processing plant that we are working with. In my point of view, the critical point is in phase 4. This is all. The critical point is in the phase 4. Okay, if we can set all the parameters in this phase, I think we can do a lot, really a lot. Answering to Geraldine, I don't see at the moment other solution than hot water, ozone. Many years ago, it was used conditioned atmosphere with carbon dioxide, but I don't think can be done anymore for safety problem. And so at the moment, we have not other efficient solution, unless I know that in the United States, they have been working in the past to work with biocontrol in storage, but biocontrol in storage is a mess even for authorization and other kind of activity. So but I'm really optimistic that if we set the sterilization, the phase 4, we can obtain very, very good results.
Okay, and another thing, Andrea, everybody know you have, in fact, you have 3 periods in the life of the fruit. The first period, it's in the orchard. The second period, we'll talk about that. It's in the plant. The name is post-harvest treatment and storage. And you have a third moment, very important. It's after the when the fresh chestnut arrive in the stores, and they sometimes they stay a long time in the stores with normal temperature 20 degrees, and the results are very, very bad. Do you think maybe it will be interesting to work to solution to preserve the quality of the fruits after the post-harvest in the plants?
Well, I think that there is a I forgot, sorry, Bertrand, I forgot to say something, that what has been presented by our colleague from France about management of the orchard is essential.
Yes.
Essential. Reducing the harvesting time, because everybody knows that if the fruit staying 24 hours more on the soil during the night, and Gnomoniopsis starting, there is nothing to do. I mean, what has been said before about the management of the orchard is essential, is part of the integrated management of the orchard. Fertilization is essential. Soil quality is essential. Density of the orchard is eventually essential. I really want to thank that Geraldine, the colleague that did the presentation, because it's a very difficult work to do, because it takes a lot of years of studying this in order to obtain result. But this is essential, Bertrand. And then if you harvest in a very short time, in a plant that I showed you before, as you bring as you take the fruits in the plant, after 1 hour they are in the sterilization sink if they have no problem of dryness. I mean, everything should be very coordinated.
Yes.
We need investment in the sector. I don't understand why with apple, pear, cherry, we have a lot of investment in equipment in post-harvest. Why not in chestnut?
Yeah.
This is my question.
And Bertrand, we need to invest in communication with the producers between us.
When yes, if we have the solutions, yes, it's important to explain, to communicate to the growers what are the solutions. But we must be sure with these solutions.
Yes. I have 2 questions for maybe Andrea and Guglielmo. Maybe answer the question that is in the heart of most of us. Why Tardif cultivars seems to be more resistant than precocious cultivars? This is the first. And the second is, what can we do with the burrs after the harvest that remain in the soil in order to help to reduce the inocle for the next year? Because this is a question that most of people ask me. What can we do? What can we do? And what about the variety?
I will answer, but maybe Guglielmo wants to say something before.
I will start with the second question about the burrs. We know that the burrs are the main source of spores. Normally, if you have a source of inoculum, the first attempt you do is to remove it. Removing them may pose questions for soil fertility and the sustainability of the cultivation of chestnut. What we are trying to do locally with the experiments that we are conducting is seeing what happens if we use the burrs to obtain compost. if we can start processes to transform the litter and reuse the litter so as to maintain soil fertility while lowering the amount of infection of inoculum. But we have also to consider not only the orchard, but what there is all around the orchard. I mean, if you have an isolated orchard or a group of orchard where everyone removes the litter, it might be effective in a first phase. But if you have an orchard that is surrounded by hectares and hectares of coppices, producing burrs, producing inoculum, this could not be effective in my opinion. Another thing we should take in consideration also for the treatment is that now we have models to predict the risk at a local scale, but to optimize treatments in the field, we should push to obtain risk models at the European scale, because treating whatever you do costs money. If you have a very low risk of infection, maybe you could optimize the treatment. And what it is done, for instance, for rice, I mean, for Piricola gavrize, just to cite an example, something that is done normal in agriculture, having an idea of what is the risk before treating. Also to estimate losses. I mean, if you have a risk of 80% of losses, maybe you are motivated to treat, or maybe you say it costs too much collecting the fruit this year. It costs too much. I don't earn enough money. I let the fruit there. So there are many further considerations to do. And I think that Andrea made a wonderful word of synthesis as of experimentation, but with his own economical effort. We should try to lobby at the European level to get funding for international and coordinated research, because otherwise it's difficult doing what Andrea rightly said, and you too. We have to uniform the treatments all around Europe to test different climate varieties and products. Otherwise, we could difficult to distinguish the role of climate, variety, chemical product, etc.
Yes.
Yeah.
And concerning the tardif, the characteristics of the cultivars, do you know something that can explain us?
I mean, maybe, but it's just an opinion. It's not based on scientific research. Different cultivars may have a different phenology, may have a different period in which flowers are receptive to the inoculum present in the air. So there might be a differential interaction with climate. This is why we need to repeat in the same place experiments accounting for climate and variety. Otherwise, we cannot distinguish the role of a putative varieties tolerance to the disease to occasional events related to the epidemiology, on just to the fact that there is a differential phenology, you know? That's my opinion.
Yeah.
I don't know if you, Andrea.
Because I put you this question because most of the collaborators, the colleagues that answered me, said, well, the first cultivars, the earlier cultivars, presented more impact, more percentage of rotten fruits than the tardif cultivars.
We should also check if in the early period there were more rain at blossoming, for instance. That could explain this fact.
I mean, basically, Guglielmo set the point that we need a collaborative project to come out of all of this.
We need
what is good now that Guglielmo, in different environment and different cultivar, there are some product that works anyway.
Yes.
And this is already a good
yeah, this is a good point, but
that we should pursue. And for the rest, I agree with Guglielmo about the birds. If you are working in organic farming, it's even more difficult to convince to remove the birds and to eventually burn the birds, because in derogation you can also burn the birds. I understand that it's difficult, but in case of heavy infection, you should anyway work like this. Eventually remove the birds before even the leaves fall down, because the birds fall down before the leaves. Remove the birds and leave the leaves go down. I know that Gnomoniopsis is also on the leaves, but it's mainly on birds and works in this way. But of course, Guglielmo demonstrated that is the main source of inoculum, so we have to work in somehow on birds.
Yeah.
Okay. Well, thank you. The discussion is very interesting, and I suggest that we continue this discussion in a small scientist group.
Yeah.
I would like to thank you for your participation. There were many of you today, which shows how important is it for the chestnut sector to find solution quickly. We look forward to seeing you maybe in Greece for the next European Chestnut Day in September. So thank you again, and have a nice end of the day. Thank you very much. Bye.
Geraldine, wait
Yes.
a moment, because we have here with us the next chairman of the EUROCASTANEA in Greece, Stefanos Diamandis.
Stefanos. Yes,
Yeah.
Stefanos, can you talk
Maybe
me
he wants to say us some words about the next meeting.
Yes, good idea.
Thank you. Thank you, José.
But Stefanos is the new chairman of EUROCASTANEA, but is one of the academically oldest scientists working on chestnut in Europe.
Thank you, Andrea. Thank you, Andrea. Before closing, Geraldine, I would like to say 1 minute. A lot of information, an enormous amount of information was presented this evening. Everybody, especially Guglielmo and Andrea, we need to help our chestnut producers. Can somebody prepare a short protocol which we can circulate to our chestnut producers so they know what to do in the coming growing season? Can somebody do that? Simple and short.
José is the responsible of this action. We will give all the information to José to prepare such.
Yes, we can try to prepare a leaflet with the protocol to treat the chestnuts coming this year, to help the farmers, to help the producers. Thank you with your suggestion.
Since I read a comment on the chat about the economic convenience of the treatment that we were exposing before, our growers in my area are doing treatment with potassium zinc phosphonate since 5 years, and eventually, if they do, it's profitable to do it.
Yes.
Otherwise, they will not do it anymore.
Yes.
Okay.
40%. Yeah, 40%, 50% is too much. And see you next times in Greece. Between 12 and 15 September, isn't it, Stefanos?
The dates are Thursday 12 to the 15th of September.
Where?
Thursday, Friday, Saturday, Sunday.
And where?
Well, we'll give you all the information.
It's not defined.
You're all welcome.
Still. Yeah, okay. Welcome. Thank you, Stefanos.
Okay. Thank you.
Thank you, Geraldine, again.
Geraldine.
Thank you. Thank you to everybody. Thank you very much.
Thank you.
Thank you very much. Bye.
Thank you.
Bye.
Bye.
Bye.